Have you ever stopped to think about why you get that flu shot every year? Or maybe how your body just knows the difference between, say, a bit of dust and a really nasty virus?
Right. It's this incredibly smart sorting system.
Yeah.
And it all really boils down to these things called antigens.
Antigens. Yeah, you hear the term maybe in medical shows or something, but what are they really?
Well, they're not just jargon. They are fundamentally like the ID cards of the biological world, the molecular signatures that let your immune system do its main job, telling friend from foe, or maybe even more deeply, telling self from non-self.
Without them, the whole immune system, as complex as it is, wouldn't know where to start.
Exactly. Wouldn't have the intel, wouldn't know what to protect or what to fight off.
Okay. So welcome to the deep dive. Today, our mission really is to get past the technical terms and dive deep into antigens. We want to figure out what they are, what makes them work, how diverse they are, and their absolutely critical role, both in protecting us and sometimes, surprisingly, in making us vulnerable.
Yeah, and we've looked at quite a bit detailed biotexts, research papers, even some illustrative medical slides. We've tried to pull out the most crucial bits, the really surprising facts for you.
Our goal is that by the end of this, you'll have a real appreciation for this molecular dance happening inside you constantly. Understanding antigens is sort of a shortcut to getting how amazing your immune system really is.
It truly is. It works silently, tirelessly.
Okay, so where do we begin? To really get antigens, I think we first need to tackle a distinction that honestly trips a lot of people up. The difference between an antigen and an immunogen, they sound so similar.
They really do. And it's a common point of confusion, even sometimes among scientists in, let's say, casual chat. They might get used interchangeably. But in immunology, they have very precise meanings, and getting that difference is key.
Okay, lay it out for us. What's the core definition of an antigen?
Right. So an antigen is basically any molecule that can do two things. First, it has to be capable of inducing an immune response. And second, this is crucial, it has to be able to react specifically with the things produced by that response.
What things? Like antibodies?
Exactly. Antibodies and lymphocytes, the specialized immune cells. So it starts the response, and it's the specific target afterwards.
It's a two-part job description, and I find this fascinating. The name itself, antigen, kind of gives it away, doesn't it? It's short for antibody generator.
That etymology is super helpful, yeah. It literally tells you its function. And you nailed the key part, specificity. Once the immune system is fired up, the antigen is like a very specific key.
Fitting into a specific lock.
Precisely. It binds with incredible precision to antibodies or to the receptors on those immune cells, the lymphocytes. That binding is immune recognition.
Okay, that's the antigen. Now, how does an immunogen differ? What's the subtle difference there?
An immunogen is, well, a bit simpler in definition. It's any substance capable of inducing an immune response, just the triggering part.
Okay, that sounds almost exactly like the first part of the antigen definition. Where's the catch?
This is it. This is the key distinction, the aha moment for many. It leads us to differentiate between two properties.
Yeah.
Immunogenicity and antigenicity.
Okay. So immunogenicity versus antigenicity. Break those down.
Okay. Immunogenicity is the ability of something to actually elicit that response, to get the B cells or T cells going. Think of it as the triggering power. Can it actually start the whole process?
Right, the power to mobilize the troops.
Exactly. Whereas antigenicity is simply the ability to combine specifically with the products of that response, like antibodies or cell receptors. It's the binding power. It can be recognized and grabbed onto, but that doesn't mean it started the fight itself.
So triggering power versus binding power. Okay, that makes sense. One starts the whole cascade. The other is just about being the target once the cascade is going.
Precisely. Which leads to this key rule in immunology. While all immunogens are antigenic, not all antigenic molecules are immunogenic.
Whoa, okay, say that again. All immunogens are antigenic.
Because if they trigger a response, the products of that response must be able to bind to them.
Right, but not all antigenic things are immunogenic.
Correct. Think of our key analogy again. You might have a tiny key that perfectly fits the lock. That's antigenicity. It can bind. But if that key is too small or maybe too flimsy to actually turn the lock mechanism by itself,
then it's not immunogenic. It fits, but it can't start the engine, so to speak.
Got it. It needs something else, maybe a bigger handle attached to it, to actually get things moving. This raises a really important question then. What does determine if a molecule has that turning power, that communogenicity? What makes the immune system actually sit up and take notice?
That's a perfect setup for our next section, because it's not just about being foreign, is it? There's a whole list of factors, almost like a checklist the immune system uses. What makes a substance truly immunogenic? Let's unpack these 7 factors.
Yeah, it's definitely a multifaceted decision. The immune system is incredibly discerning. Let's start with the first factor, molecular size.
Okay, so does size matter here? Is bigger generally better for getting the immune system's attention?
Generally, yes. Larger molecules tend to be much better immunogens. The really potent ones are often large proteins, typically over, say, 100,000 Daltons in molecular weight. That's quite large on a molecular scale.
And then on the flip side, what about small molecules?
Molecules smaller than about 5,000 Daltons are usually not immunogenic on their own. They're just too small to effectively bridge receptors or trigger a strong signal. But, and this is really clever and used a lot in medicine, you can make them immunogenic.
How? By sticking them onto something bigger.
Exactly. You chemically attach these small molecules, which we'll later call hadrons, to larger carrier particles. These could be inert substances like bentonite or kaolin in a lab setting, or, more commonly in vaccines, larger carrier proteins.
So you're giving this tiny molecule a big visible flag.
That's a great way to put it. This manipulation is vital. Imagine trying to make a vaccine against just a single sugar molecule from a bacterium's surface on its own. The immune system might completely ignore it, attach it to a big protein. Now it gets noticed.
Clever. Okay, so size is one factor. What's next? I assume where it comes from matters foreignness, the whole self versus non-self thing.
Absolutely fundamental. This is maybe the core task of the immune system. For a molecule to trigger a response, your immune system must recognize it as non-self.
How does it know what's self?
Through a process called tolerance, which mostly happens early in development. The immune system basically learns to recognize and ignore the body's own components. If it didn't, well, it would constantly attack itself.
Which would be autoimmune disease, right?
Yeah.
We'll get to that. Does the degree of foreignness matter?
It does. Generally, the more evolutionarily distant two species are, the more immunogenic a molecule from one will be in the other. So, a protein from a cow is likely to trigger a much stronger response in a human than a protein from, say, a chimpanzee, because we're much more closely related to chimps.
Makes sense. But you mentioned it. What happens when that tolerance, that ability to ignore self, breaks down?
That's exactly the basis of autoimmune diseases. The system makes a mistake. It identifies some part of the body, maybe a protein, a cell type, as foreign, and it launches an attack. Conditions like rheumatoid arthritis, type 1 diabetes, lupus, they're all consequences of this breakdown in self-tolerance. The protector becomes the attacker.
A devastating error in recognition. Okay, third factor. Chemical structural complexity. Are simple molecules as good at triggering a response as complex ones?
Not usually, no. There's a definite hierarchy. Proteins with their complex folding and diverse amino acid sequences are generally the best immunogens. They offer lots of unique shapes and chemical properties for the immune system to recognize.
What about other types of molecules, like sugars or fats?
Carbohydrates, like polysaccharides, are typically weaker. And simple lipids and nucleic acids, like DNA or RNA, are generally poor immunogens on their own. However, and this is important, their immunogenicity can be dramatically boosted if they're combined with proteins. Think glycoproteins or lipoproteins. That combination adds complexity.
So complexity is key. It gives the immune system more distinct features to grab onto.
Exactly. And even within proteins, complexity matters. A simple repeating chain of the same amino acid, a homopolymer, is much less immunogenic than a heteropolymer, which is made of different amino acids and can fold into more intricate shapes.
Is there any protein that breaks this rule? A complex molecule that isn't very immunogenic?
There's a classic example, gelatin. Even though it's a protein derived from collagen, it's surprisingly non-immunogenic.
Why is that?
It's mainly due to its structural instability and lack of a fixed complex three-dimensional conformation. It's kind of floppy and repetitive, structurally speaking, making it hard for immune receptors to get a consistent, strong grip.
Fascinating. So stable, complex shape is important. What about factor 4? Stability and degradation. Does the antigen need to stick around for a while?
Yes, but it's a balance. For a substance to act as an immunogen, it generally needs to be stable enough to persist in the body long enough for the immune system to encounter it. But crucially, it also needs to be susceptible to being broken down or metabolized by the body's enzymes.
Why does it need to be broken down? Wouldn't you want the target to stay intact?
Well, here's how it works for many responses, especially involving T cells. Antigens get picked up by specialized cells like macrophages or dendritic cells. We call them antigen-presenting cells, or APCs. Inside these APCs, the antigen is chopped up into smaller pieces, smaller peptide fragments.
And those fragments are what get shown to the immune system.
Exactly. These fragments contain the specific hotspots, the epitopes that T cells recognize, but only when they're presented on special molecules called MHC, which we'll get to. So the antigen needs to be digestible, if you will.
So if something is completely indigestible, like maybe certain plastics, or if it breaks down too fast,
Then it's generally not immunogenic. Things that are totally insoluble or broken down almost instantly, or made of synthetic components our enzymes can't handle, like D-amino acids instead of the natural L-amino acids. They usually fail to trigger a response because they can't be properly processed and presented.
it's like biological processing is required. Okay, factor 5. The biological system itself, the host factors, does everyone react the same way?
Absolutely not. This is where genetics really comes into play. What's immunogenic in one person might not be in another. We talk about responders and non-responders to certain antigens.
What determines that difference?
It often comes down to variations in genes that control key parts of the immune system. Genes for the B cell and T cell receptors themselves, the molecules doing the direct recognition, and, really importantly, genes for the major histocompatibility complex, the MHC molecule.
Those are the molecules that present the antigen fragments to T cells.
Precisely. Your specific set of MHC molecules determines which peptide fragments can be effectively presented to your T cells. So, if you don't have an MHC molecule that can bind and present a key fragment from a particular antigen, your T cells might never get properly activated, and you might be a non-responder to that specific antigen.
So your personal genetic makeup dictates your immune responsiveness to some extent. That helps explain why, say, vaccine effectiveness can vary a bit between people.
Exactly. Or why some people are more susceptible to certain infections or even autoimmune conditions. It's your unique immune landscape.
Right. Factor 6, dosage and route of administration. This seems straightforward, how much antigen you get and how it gets into you.
It is, but with some nuances. Both the dose and the route matter significantly. You might think more is always better, but that's not true for immunogenicity.
Oh, too much can be bad.
Yes. Very low doses might simply be insufficient to stimulate enough immune cells to get a response going. But paradoxically, extremely high doses can sometimes lead to a state called immunological paralysis or high-dose tolerance.
Paralysis. The immune system just gives up.
Kind of. It's a form of tolerance, where the system gets overwhelmed by the sheer amount of antigen and actively shuts down the response, possibly mistaking it for self.
Yeah.
Because it's so abundant. So there's usually an optimal dose range.
A sweet spot. And what about the route, like swallowing versus injection?
Big difference. Parenteral administration, things like intramuscular or subcutaneous injections, basically bypassing the gut, generally induces a much stronger immune response than, say, oral or nasal routes.
Why is that?
Injections often deliver the antigen more directly to lymph nodes and tissues rich in immune cells, allowing for efficient uptake by APCs. Oral routes face challenges like digestion in the stomach and intestines, and also the gut has specialized tolerance mechanisms to prevent unwanted reactions to food antigens. That's why most vaccines are injected to ensure a robust response.
Makes practical sense for vaccine design. Okay, and that leads perfectly to the 7th and final factor. Adjuvants, the immune boosters. These are crucial for vaccines, right?
Absolutely crucial. Adjuvants are substances that you mix with an antigen and inject together, and they dramatically enhance the immune response to that antigen. Importantly, they do this without changing the antigen structure itself.
So they're like cheerleaders for the immune system, making it react more strongly to the main event.
That's a great analogy. They work in several ways. Some, like aluminum salts, often called alum, which is in many common vaccines, create a kind of depot at the injection site. They make the antigen hang around longer, slowly releasing it, giving immune cells more time to find and react to it.
Prolonging the exposure.
Exactly. Others work by directly stimulating immune cells. They might trigger pattern recognition receptors on APCs, basically sounding an alarm that says danger. This makes the APCs mature faster, migrate to lymph nodes more effectively, and present the antigen much more powerfully to T cells.
So they're not just keeping the antigen around, they're actively kicking the immune cells in a high gear.
Yes. They can increase the total amount of antibody produced, help induce cell-mediated immunity, the T cell side, and generally just make the whole response stronger and longer lasting. Besides alum, other examples include oil and water emulsions or components derived from bacteria. They are often the unsung heroes in making vaccines effective, allowing us to use smaller amounts of antigen but still get really good protection.
Really fascinating how we can manipulate the system like that. Okay, so we know what antigens are and what makes them trigger a response. Now let's zoom in. How does the immune system actually see these antigens? It's not the whole molecule, right? You mentioned epitopes.
Exactly. Antigens, especially big proteins, are complex landscapes. But the B cells and T cells, the lymphocytes doing the recognizing, don't grab onto the whole thing. They focus on specific, small, discrete sites on the antigen. These are the antigenic determinants, or more commonly, epitopes.
So the epitope is the specific little bit, the molecular handle that the immune cell receptor actually binds to.
Perfect. That's exactly it. It's the immunologically active region that directly interacts with the antigen-specific receptors on lymphocytes or with secreted antibodies.
And you mentioned the lock for this key before, the paratope.
Right. The paratope is the corresponding site on the antibody molecule or the T cell receptor that physically binds to the epitope. It's shaped to be perfectly complementary. This incredibly precise epitope-paratope interaction is what gives the adaptive immune response its remarkable specificity.
One lock, one key. Got it. Now, do B cells and T cells look for the same kinds of epitopes, or are there differences there? It feels important.
Huge difference. It's fundamental to how the two main arms of adaptive immunity work. Let's start with B cell epitopes. These can be formed in two main ways. They can be sequential or linear, meaning they're defined by a specific sequence of amino acids right next to each other in the protein chain.
Like a specific word in a sentence?
Kind of. Or they can be conformational. These are formed by the protein folding up into its 3D shape, bringing amino acids that might be far apart in the linear sequence close together in space. This creates a unique surface patch that the B cell recognizes.
So the 3D shape itself creates the epitope.
Correct. And a key thing about B cells is that their receptors, which are basically membrane-bound antibodies, can recognize these epitopes directly on intact, folded proteins, or even on polysaccharides or lipids, often floating freely in the blood or lymph. They don't need the antigen to be processed first.
Okay, so B cells are like surface scanners looking for intact shapes. But T cell epitopes are different. You said they need processing and presentation.
Exactly. This is where it gets really interesting and a bit more complex. T-cell epitopes are almost always linear peptides, typically quite short, maybe 8 to 15 amino acids long. And the absolute crucial point is T-cells cannot recognize these peptides on their own.
They need that MHC molecule you mentioned.
They only recognize these peptide epitopes when they are bound and presented by major histocompatibility complex, MHC molecules, on the surface of another cell, usually an antigen-presenting cell, APC, or an infected body cell.
So the antigen gets chopped up inside a cell, a little peptide piece gets loaded onto an MHC molecule, and that whole MHC peptide complex is then displayed on the cell surface for a T cell to inspect.
Precisely. T cells don't see free-floating antigens like B cells do. They survey the surfaces of other cells, looking for these MHC-presented peptides. It's a mechanism perfectly designed to detect intracellular problems, like viral infections or cancer, where abnormal proteins are being made inside the cell.
And there are different MHC classes, right, for different types of T cells?
Yes, MHC class I molecules are found in almost all our nucleated cells. They primarily present peptides derived from proteins made inside that cell. These are recognized by CD8 plus cytotoxic T cells, the killer T cells. If a cell presents a viral peptide on MHC class I, a CD8 plus T cell recognizes it as infected and kills the cell.
Okay, MHC class 1 is for intracellular threats, recognized by killer T cells. What about MHC class 2?
MHC class II molecules are normally found only on specialized APCs, macrophages, dendritic cells, B cells. They present peptides derived from proteins that the APC has engulfed from outside, like bacteria or bacterial toxins. These are recognized by CD4 plus helper T cells.
And helper T cells don't kill directly.
No, they orchestrate. When a helper T cell recognizes an antigen on MHC class II, it gets activated and releases signals, cytokines, that help activate B cells to make antibodies, help activate macrophages to kill engulfed pathogens more effectively, and help activate cytotoxic T cells. They're the master regulators.
Wow. So MHCI for inside threats, MHC2 for outside threats, guiding the right kind of T cell response. That's elegant. And this MHC restriction explains why T cells generally don't respond well to things like polysaccharides.
Exactly. Those types of molecules generally aren't processed into peptides and loaded onto MHC molecules effectively. T cells primarily see peptides. This is why proteins are often called T-dependent antigens. You need T cell help, triggered by MHC-presented peptides, to get a really strong, full antibody response with memory.
That's a critical piece of the puzzle. Now, this system is incredibly specific, but can it ever get confused? What about cross-reactivity, when an antibody or T cell for one thing reacts with something else?
Yes, that definitely happens. Sometimes an antibody or T cell receptor that was generated against one specific epitope might bind, maybe a bit less strongly but still significantly, to a different but structurally similar epitope on an unrelated antigen.
Why would that happen? Just random similarity.
Often, yes. Two different proteins, maybe from completely different sources, might just happen to have a small region that looks very similar chemically or structurally. This cross-reactivity can sometimes be useful, like in the diagnostic tests we might discuss later, but it can also cause problems.
Like false positives in lab tests.
That's one possibility. But more seriously, it can be a major mechanism behind some autoimmune diseases. The immune response starts against a foreign invader, like a bacterium, but then mistakenly attacks the body's own tissues because some molecule on those tissues looks too much like the bacterial antigen. We call that molecular mimicry.
Okay, we'll definitely come back to that. So, understanding epitopes and how they're recognized is key. Now, let's explore the sheer variety. You've hinted at different types. Let's go through this diverse gallery of antigens.
Right. It's useful to classify them. First, the straightforward ones. Complete antigens. These are the substances that can do it all. They're immunogenic, trigger a response, and antigenic, react specifically with the products. They effectively stimulate antibody formation and give a measurable reaction. They're the full package.
Okay. Then you mentioned those incomplete ones earlier, haptens. Remind us about those.
Haptens are fascinating. They are typically small organic molecules, low molecular weight. The key thing is, they are antigenic, meaning if antibodies already exist, they can bind to them. But they are not immunogenic on their own. They cannot trigger an immune response by themselves.
Too small or simple.
Exactly. But, and here's the clinically crucial part, they become immunogenic if they chemically attach or covalently couple to a larger carrier protein.
So the hapten provides the specific shape, the epitope, but the carrier provides the necessary size and complexity to get the immune system's attention.
Precisely. The hapten carrier complex as a whole is now large enough and complex enough to be immunogenic.
And the classic example you gave was penicillin.
Penicillin is the perfect example. For most people, it's fine. But in allergic individuals, penicillin acts as a hapten. It can spontaneously bind to the body's own proteins, like serum albumin. This creates a penicillin protein complex that is immunogenic. The immune system sees this altered self-protein as foreign, mounts a response against the penicillin part, and bam! You get an allergic reaction, sometimes severe. Understanding haptens is key to understanding drug allergies.
A small molecule causing big problems by hitching a ride. Got it. Okay, next type. T-cell dependent, TD antigens. These are the ones needing T-cell help.
Yes, the vast majority of protein antigens fall into this category. To get a really strong B-cell response, meaning lots of antibodies, switching to different antibody types like IgG or IgA, and developing long-lasting memory, these antigens require interaction between B cells and activated T helper cells.
What kind of antigens are typically TD?
Things like proteins from viruses, bacteria, foreign to blood cells, serum proteins, basically, most complex protein structures. The response they generate is characterized by that full range of antibody types, isotypes, increasing antibody binding strength over time, affinity maturation, and, crucially, the formation of immunological memory. This memory is why vaccines containing TD antigens give long-term protection. Your body remembers the antigen.
So TD antigens, strong response plus memory. What about the opposite? T-cell independent, TI antigens.
These are antigens that can activate B cells directly without help from T helper cells. They have a different way of stimulating the B cell.
How do they do that?
TI antigens often have highly repetitive structures. Think of large polysaccharide molecules from bacterial capsules or lipopolysaccharide, LPS, from gram-negative bacteria. These molecules have many identical epitopes arranged close together. This allows them to physically cross-link many B-cell receptors on the surface of a single B-cell simultaneously.
Like pushing many buttons at once.
Exactly. That strong simultaneous signaling can be enough to activate the B-cell directly without the secondary signals usually provided by T-cells.
What's the response like then? Is it as good as for TD antigens?
Generally, no. The response to TI antigens tends to be quicker but weaker. It primarily consists of IgM antibodies. There's little or no class switching to IgG or IgA, minimal affinity maturation, and crucially, very little immunological memory.
So you might clear the infection, but you won't necessarily have a long-lasting immunity.
Correct. This is one reason why immunity to some encapsulated bacteria, where the capsule is a major TI antigen, might not be as durable as immunity to viruses where protein antigens dominate.
That's a really important distinction for vaccine strategy, too, I imagine. Okay, moving on to something that sounds dramatic. Super antigens. What makes them super?
They're super in their ability to activate T cells, but in a very dangerous, non-specific way. These are usually proteins produced by certain bacteria, like Staphylococcus aureus, causing toxic shock syndrome, or viruses.
How do they activate T cells differently?
They completely bypass the normal, exquisite specificity of T cell recognition. Remember how T cells usually only recognize a specific peptide presented by a specific MHC molecule?
Right, the lock and key.
Superantigens ignore that. They act like a molecular clamp. They physically link MHC class II molecules on APCs directly to the T cell receptor, but they bind outside the normal peptide binding groove and to regions of the T cell receptor shared by many T cells, the VE domain.
So they're forcing a connection between the APC and the T cell, regardless of the specific peptide involved.
Exactly. They short-circuit the system. Because they bind to common regions on T cell receptors, they can activate a huge proportion of the body's T cells simultaneously.
How huge are we talking?
A normal antigen might activate, say, 0.0001% to 0.01% of your T cells, only those specific for its epitopes. A superantigen can activate maybe 5%, 10%, even up to 20% of all your T cells at once.
That sounds bad, like a massive overreaction.
It is. This leads to an enormous sudden release of inflammatory cytokines from all those activated T cells, what's often called a cytokine storm.
And that causes the symptoms.
Yes, this cytokine storm causes widespread inflammation, fever, rash, plummeting blood pressure, shock, and potentially organ failure. It's the mechanism behind toxic shock syndrome and certain types of food poisoning. The immune response itself becomes the major threat.
A terrifying example of immune activation gone wrong. Okay, let's shift gears to something more personal. Histocompatibility antigens. You mentioned MHC before. These are the same thing, essentially, often called HLA in humans.
Yes, precisely. MHC stands for Major Histocompatibility Complex, which refers to the genes. HLA stands for Human Leukocyte Antigen, referring to the protein molecules produced by those genes in humans. These are the molecules found on the surface of almost all your cells that act like your body's unique ID card.
Unique to each individual.
Highly unique. The HLA genes are incredibly diverse in the human population. There are thousands of different versions or alleles. So the specific combination of HLA molecules on your cells is like a fingerprint, very unlikely to be identical to someone else's unless you're an identical twin.
And their main job, immunologically, is presenting those peptide fragments to T cells.
That's their critical role in adaptive immunity, yes. As we discussed, MHC class I presents peptides from inside the cell to CD8 plus killer T cells, and MHC class II presents peptides from engulfed material to CD4 plus helper T cells. This presentation is absolutely required for T cell activation.
And this uniqueness is why they are so crucial in transplantation.
Absolutely paramount. If you transplant an organ, say a kidney, from one person to another, the recipient's T cells will recognize the donor's HLA molecules, which are different from their own, as foreign. This triggers a powerful immune attack against the transplanted organ, leading to graft rejection.
So HLA matching between donor and recipient is vital.
It's the single most important factor for transplant success. The closer the HLA match, the lower the risk of rejection. Even with good matches, recipients usually need immunosuppressive drugs to help prevent their immune system from attacking the foreign but life-saving graft.
It highlights how intensely the immune system guards that self-identity defined by HLA. Which brings us neatly to autospecificity and autoimmunity. Normally, our own stuff, our self-antigens, should be ignored. But sometimes
that tolerance fails. And there are several ways this can happen. One involves sequestrated antigens.
Sequestrated, like hidden away.
Exactly. Some parts of the body, like the proteins inside the lens of your eye, or sperm cells within the testes, or myelin proteins insulating nerves, are normally anatomically isolated. The developing immune system never really sees them properly during the tolerance induction phase.
So no tolerance is built up against them.
Correct. They're immunologically privileged or ignored. But if injury or disease breaks down that barrier, say, trauma releases lens protein into the bloodstream, or a vasectomy allows sperm antigens into the circulation, the immune system might encounter these hidden self-antigens for the first time.
And sees them as foreign.
And mounts an immune response, leading to autoimmune attack on the eye, or testes, or nerves. Autoimmune uveitis or multiple sclerosis might involve elements of this.
Wow. What about antigens that aren't hidden, but change somehow? Neoantigens or altered antigens?
Right. Your own cell's molecules can be altered. This might happen due to things like irradiation, certain drugs, chronic inflammation, or viral infection. A virus, for instance, might integrate its genes, causing the cell to produce viral proteins, or it might modify existing host proteins. Bacterial enzymes might also chemically alter host tissues.
So the cell's ID card gets changed.
In a way, yes. These altered molecules, or neoantigens, might no longer be recognized as self by the immune system. The immune response mounted against them can contribute to autoimmunity if the alteration is widespread on normal tissues. Or, excitingly, in cancer, these neoantigens, often caused by mutations, can become targets for immunotherapy, helping the immune system specifically find and kill cancer cells.
And then there's that idea you mentioned earlier, the really sneaky one, molecular mimicry.
Yes, this is a fascinating and major cause of autoimmunity. It's when a foreign antigen, like from a bacterium or virus, happens to look structurally very similar to one of your own self-antigens.
A case of mistaken identity.
Exactly. The immune system mounts a perfectly normal, strong response against the invading pathogen. But the antibodies, or T cells, produced during that response also recognize and attack the similar-looking self-antigen on your own tissues.
And the classic example is rheumatic fever after strep throat.
That's the textbook case. The M proteins on Streptococcus pyogenes resemble proteins in the human heart valves, joints, and even brain. So the anti-strep antibodies cross-react, causing inflammation and damage in those tissues long after the infection is gone. It's the immune response itself, triggered by mimicry, that causes the disease.
A truly unfortunate consequence of structural similarity. Okay, a couple more classifications. What about organ specificity?
This refers to the fact that certain antigens are characteristic of a particular organ, and interestingly might be shared across different species for that same organ. For example, brain tissue from different mammals might share certain brain-specific antigens. Kidney tissue might share kidney-specific antigens across species, and so on.
And heterophile specificity, that sounds even broader.
It is. Heterophile antigens are even stranger. They are similar or identical antigens found in tissues of biologically distant species, sometimes even across different classes or kingdoms. So, an antigen in a bacterium might be very similar to one on a sheep red blood cell, for instance.
Why is that significant?
Because the antibodies produced against one can cross-react with the other. And this weird cross-reactivity is actually exploited in some classic diagnostic tests.
You mentioned this earlier, like the Weil-Felix test.
Exactly. The Weil-Felix reaction for rickettsial infections uses protease bacteria antigens because antibodies against rickettsia happen to cross-react with them. The Paul-Bunel test for infectious mononucleosis uses sheep red blood cells because antibodies against the Epstein-Barr virus cross-react with an antigen on those cells. Cold agglutination tests for mycoplasma pneumonia work similarly. We use these quirky cross-reactions as diagnostic shortcuts.
Very clever use of a biological oddity. Finally, in this gallery, let's talk about tumor antigens. Hugely important for cancer.
Absolutely. This is a massive field in cancer research and therapy. Tumor antigens are molecules present on cancer cells that distinguish them from normal cells. Ideally, the immune system can recognize these antigens and attack the tumor.
Are they completely unique to the tumor?
Sometimes yes, sometimes no. We divide them into two main types. Tumor-specific transplantation antigens, TSTAs, are truly unique to the tumor cells, often resulting from the specific mutations that cause the cancer. They aren't found on normal cells at all. Different chemically induced tumors might have unique TSTAs. Interestingly, tumors caused by the same virus might share TSTAs derived from that virus.
So potential targets specific only to the cancer. What's the other type?
Tumor-associated transplantation antigens, TADAs. These are antigens found on tumor cells, but they might also be found on some normal cells, although often at much lower levels, or perhaps only during certain developmental stages, like fetal development. These include things like certain carbohydrate structures that change on cancer cells, differentiation antigens like CD10, seen in some leukemias, or PSA, prostate-specific antigen, and importantly, oncofetal antigens. These are proteins like AFP, alpha-fetoprotein, and CEA, carcinoembryonic antigen, that are normally produced mainly by the fetus, but get re-expressed by certain adult cancers, like liver or colon cancer.
And these TADAs, even if not totally unique, are useful.
Usually useful as biomarkers. Detecting elevated levels of PSA, AFP, or CEA in the blood can help diagnose certain cancers, monitor how well treatment is working, or detect if the cancers come back. Even if the immune system doesn't always eliminate tumors based on TADAs, their presence is diagnostically invaluable.
Okay, that's a comprehensive tour of antigen types. Now, let's focus specifically on antigenic structures in microbes. This is where the immune system meets the invaders head-on. How do bacteria present themselves antigenically?
Bacteria have a whole array of antigens, many linked to their structure and virulence. A major one is the bacterial capsule, if they have one. This is usually a slimy, gelatinous layer, often made of polysaccharides, surrounding the bacterium. Think Streptococcus pneumonia or Haemophilus influenza.
What does the capsule do antigenically?
Well, the specific sugars in the capsule are major antigens that define the bacterial serotype, like different strains. But functionally, the capsule is often a key virulence factor because it helps the bacterium evade phagocytosis. It makes it slippery and hard for immune cells like macrophages to grab onto. Antibodies against the capsule can be protective, but sometimes the capsule is only weakly immunogenic or masks other antigens.
So it's an ID tag and a shield. What about the cell wall itself?
The cell wall is rich in antigens. In gram-positive bacteria like Staph aureus, components like teichoic acids are important antigens and can help the bacteria stick to host cells. In gram-negative bacteria, the outer membrane contains that lipopolysaccharide, LPS we mentioned. The long polysaccharide chains sticking out from LPS are called O antigens. These are major surface antigens, highly variable between strains, used for serotyping, like E. coli O157, and are very potent stimulators of the immune system. The lipid part of LPS, lipid A, is the endotoxin that causes septic shock.
Okay, capsule cell walls. What about things bacteria use to move?
Flagella. If a bacterium has flagella for swimming, the protein building blocks of the flagella, called flagellin, act as flagellar H antigens. These are also highly antigenic and variable, used for serotyping, like the H in E. coli O157, and remember phase variation. Some bacteria, like Salmonella, can switch between expressing different types of H antigens, helping them evade antibodies targeting the previous type.
Constantly changing their license plate. Any other key bacterial surface antigens?
Lots. Things like pili or fimbriae, hair-like structures for adhesion, various outer membrane proteins, OMPs, in gram-negatives, proteins like the M protein in Streptococcus pyogenes, involved in virulence and linked to rheumatic fever. And some bacteria, like Salmonella typhi, have an additional surface polysaccharide layer called the Vi antigen, which overlays the O antigen and further helps inhibit phagocytosis and immune recognition. Bacteria have many ways to present themselves antigenically, often tied directly to their ability to cause disease.
It's a complex surface they present. What about viral antigens? They're simpler structures than bacteria, right?
Structurally simpler, yes, but their antigens are just as crucial. Viruses have internal antigens, like the nuclear protein that packages the viral genome, or matrix proteins that provide structure. These are often conserved within viral types and can be targets for T cells, especially cytotoxic T cells recognizing infected cells. But the really key ones for immunity and infection are the surface antigens.
Like the spikes we often see in diagrams.
Exactly. Think of the hemagglutinin, HA, and neuraminidase, NA proteins on the surface of influenza viruses, or the spike S protein on coronaviruses, or various glycoproteins, GP, on the surface of HIV, like GP120 and GP41.
What do these surface antigens do?
They are critical for the virus life cycle, especially for attaching to and entering host cells. HA binds to sialic acid on our cells. The SARS-CoV-2 spike protein binds to ACE2. HIV GP120 bonds to CD4. Because they're on the surface, they're also the primary targets for neutralizing antibodies that can block the virus from infecting cells in the first place. Their structure determines viral classification and tropism, which cells they infect.
And this is where antigenic variation in viruses comes in, like the flu.
Precisely. Viruses, especially RNA viruses like influenza, are notorious for it. Antigenic drift refers to small, gradual changes in the HA and NA genes due to point mutations that accumulate over time. This causes small changes in the surface proteins, meaning antibodies from previous infections or vaccinations might not bind as well.
So immunity wanes gradually.
Yes. But then there's antigenic shift. This is a much bigger, more abrupt change, usually happening only in segmented viruses like influenza. If two different flu strains infect the same cell, say a human strain and an avian strain in a pig, their gene segments can get reshuffled, creating a completely new virus with a novel combination of HA and NA.
And if humans have no pre-existing immunity to this new combination?
That's when you can get pandemics.
Ugh.
The 1918 Spanish flu, the 1957 Asian flu, the 1968 Hong Kong flu, the 2009 H1N1 swine flu, these were all caused by antigenic shift events. This constant evolution, drift, and shift is why we need updated flu vaccines frequently and why developing universal long-lasting vaccines is so challenging really is an evolutionary arms
A
race.
constant race indeed. Okay, so we've covered the types, the structures. Let's pull it all together. What is the central role of antigens in the overall immune response? They're the trigger, right?
They're absolutely central to the entire acquired or adaptive immune system, the specific memory-forming part. They're the initial signal, the thing that gets recognized
By which cells again?
by specific receptors on B lymphocytes and T lymphocytes. Each B or T cell has receptors that are essentially unique to it, designed to recognize one specific epitope. When that lymphocyte encounters its matching antigen,
That's the trigger for activation.
that's the trigger. It sets off a cascade. The lymphocyte gets activated, starts dividing rapidly, clonal expansion, and differentiates into effector cells ready to fight, and also into memory cells for long-term protection. The whole specific response hinges on that initial antigen recognition.
Let's recap the B cell pathway quickly. B cell sees its antigen.
It gets activated, often with T cell help for TD antigens, proliferates, and becomes a plasma cell. Plasma cells are antibody factories, churning out large amounts of antibodies specific for that original antigen.
And those antibodies go out and do what?
They circulate in blood and lymph. When they find the antigen, they bind to it. This binding can neutralize the antigen, block its function, opsonize it, tag it for phagocytosis by macrophages, or activate the complement system, a cascade of proteins that pokes holes in bacteria or recruits other immune cells.
Yeah.
Basically, antibodies target the antigen for elimination.
Okay, that's the antibody-mediated humoral response. Now, the T cell pathway. T cell sees its antigen presented on MHC.
Right. If it's a CD4 plus helper T cell recognizing antigen on MHC class 2, usually on an APC that engulfed something foreign, it gets activated. It then releases cytokines, chemical messengers that, as we said, amplify the entire immune response. They help activate B cells, help activate macrophages, help activate CD8 plus T cells. They're the conductors of the immune orchestra.
And if it's a CD8 plus cytotoxic T cell recognizing antigen on MHC class I, on any infected or cancerous body cell.
It gets activated, often needing help from CD4 plus cells too, and becomes a killer. It seeks out other body cells, presenting that same specific antigen on MHC class 1, latches onto them, and delivers a lethal hit, inducing apoptosis programmed cell death in the target cell. This eliminates infected cells before viruses can replicate or kills potentially cancerous cells.
So direct cellular killing. It's really this two-pronged approach, antibodies for extracellular threats, killer T cells for intracellular threats, all coordinated by helper T cells, and all initiated by recognizing specific antigens.
That's the beauty of it. This highly specific interaction allows the immune system to mount a tailored attack against virtually any foreign invader or abnormal cell it encounters, while ideally leaving the trillions of healthy cells unharmed. It all hinges on that initial specific recognition of the antigen.
We've certainly covered a lot of ground. From just defining an antigen to understanding all those factors that make something trigger a response, the difference between triggering and just binding, the specific epitopes, the incredible diversity of antigen types happens, superantigens, tumor antigens, and how microbes use them. It's incredibly intricate.
It really is. And understanding this isn't just academic biology. It underpins so much of modern medicine. Vaccines, they work by presenting antigens in a safe way to generate memory. Allergies, that's an inappropriate immune response to otherwise harmless environmental antigens, often involving happens or cross-reactivity. Autoimmune diseases, the tragic failure to distinguish self-antigens from foreign ones.
Transplant medicine relies entirely on understanding HLA antigens. And even cancer therapy is increasingly focused on targeting tumor antigens or boosting the immune system's ability to see them.
Exactly. Having even a basic grasp of antigens gives you, the listener, a much deeper appreciation for what your body is doing constantly, silently to protect you. It's an ongoing surveillance and response system of incredible sophistication.
That constant vigilance, built on this complex language of molecular recognition, it's truly a marvel. So maybe a final thought to leave everyone with. If antigens are the words that the immune system reads, and our bodies are constantly learning new dialects of these words from every cold, every infection, every vaccine we encounter, what does that lifelong molecular education mean? How does it shape our individual immunity over time, and what could it mean for how we engineer immunity in the future? What does it really mean to be well informed by the very molecules that both define us and defend us?